Okay, then. Good morning, everybody. And we're going to carry on our discussion about the immune system today. And what I want to emphasize, this is we're only again kind of doing a broad outline skeleton of the topic because it is extremely complex. But again, it's really important that, especially in this time of the pandemic, that we get some good understanding about the rudiments, at least, of how the immune system works. And we ended up last time, we talked mainly about the innate immune system, which is like this kind of pre-programmed part of the immune system that responds extremely quickly. And it has to be said very, very efficiently in most cases. When we are exposed to infection, especially of things like bacteria and fungi, the innate immune system is really strong enough to take care of it. But today we're going to go on and talk about the other arm of the immune system, which is the adaptive immune system. So I just want to remind you about what we talked about, because there is a link between the innate immune system and the adaptive immune system. Remember what we said about this portion of the innate immune system, which is produces the classic inflammatory response. And that inflammatory response is largely generated by the production, first of all, by the recognition by cellular elements of the innate immune system, especially macrophages, mast cells, those kinds of cells, that some kind of insult has happened. There's been damage, there are pathogens present, etc., etc. When that happens, the macrophages especially produce a set of immune hormones called the cytokines, which are released and which into circulation, and which cause all sorts of responses by the immune system. They galvanize the neutrophils, for example. They call the neutrophils to the site of infection. They get the body to produce more neutrophils. They raise the metabolic rate locally, and they may raise it globally as well, etc., etc. These are all the classic symptoms of inflammation. And if we to boil inflammation right down to its essential, its most classic effect of these hormones is on blood vessels, is to cause them to dilate, to bring lots of blood in, and to bring in the cells of the immune system, which will operate to try and get rid of the infection. Now, as I told you, most of the time, this is sufficient. The innate system is very powerful and can deal with many of these situations. But there are times and kinds of insult which the innate immune system finds difficult to deal with efficiently and to stop entirely. And those are things especially like viral infections, those sorts of things. We certainly get strong innate immune responses to viral infection, but the innate response is seldom able to get rid of viral infection. I'll just use viral as an example because that is so pertinent to what we're talking about now, with the pandemic and everything else. So in talking about this kind of localized event here, there is a cell which I've left out, which is tremendously important. It's a cell which rather like the macrophage. I told you before, in circulation, we see those cells called the monocytes. The monocytes leave circulation and they enter the tissues. They have two fates. One is to form, that some of them form these macrophages, which carry on moving around the tissues as sentinels. The other fate of the monocyte is to form a cell called a dendritic cell. Dendritic cells also make their way through the tissues. They wander around, and they are also surveillance cells. But the difference there is that when a dendritic cell encounters things like pathogen, or when it, by the way, can recognize presence of bacteria, it can also recognize the presence of viruses. When it does so, instead of hanging around this area here during this localized inflammation response, the dendritic cell begins to migrate. The dendritic cell will leave this area and will go to a lymphatic vessel and begin to make its way to a lymph node. And it's going to the lymph node with information that it has gathered here. It will have taken up pathogen of whatever kind it's recognized, and it'll carry that information from this area here to the lymph node to communicate with other cells in the lymph node. So just remember this, that we're going to follow this whole story about the dendritic cell and about the reactions that occur around the dendritic cell. They're rather complicated, but we'll try and boil it down to a story that is relatively easy to understand. Okay, so before I leave the topic of inflammation, I just want to point out to you again, I did mention this, that these inflammatory effects can be local, right around where the infection occurs, or they can become global for the whole body. And one of the really important sets of hormones released, for instance, by the macrophages, causes the hypothalamus of the brain to rack up our metabolic rate and to raise our temperature. That is where the temperature comes from when you have an infection. If you have a severe bacterial infection, you'll get the same thing, but we very quickly develop it sometimes with a viral infection as well. And this is an inflammatory effect, but it's yes, go ahead.
So isn't it like when you have like a fever or something, it's because it's your body's way of telling you that the pathogens in your body are being fought or fighting off the virus that's in you or the bacteria?
It's a very good sign, yes, that your immune system is reacting to something. Yes, but it's actually useful, especially for viral infections, because viruses don't function at high temperature.
I was just about to say that also, yeah, because I've also noticed that when we get like fevers and stuff, it's also because, again, viruses don't usually want to live so much on higher temperature surfaces.
Correct. That is correct.
Our body naturally raises our temperature.
Yes, and our immune cells and antibody and everything work very well at a slightly higher temperature. So it gives us an advantage. The problem is, if you raise the temperature too high, then the hypothalamus loses control. The hypothalamus loses its ability to regulate body temperature. And then the body temperature just starts to go higher and higher and higher. So a moderately high temperature is beneficial. Too high a temperature is very dangerous, but it's very seldom that happens. Usually we develop a temperature and it just hangs at around 101 degrees Fahrenheit or so, and it doesn't go much higher. This is also, it raises the question, is it a good idea, for instance, when you're getting a cold or if you are unlucky enough to have flu, is it a good idea to take things which will lower your temperature? Unfortunately, having the high temperature is unpleasant. It feels bad. But many of the symptoms that you feel, the aching, remember I told you that one of the effects of the cytokines is to increase your sensitivity to pain, increases the sensitivity of nerve endings. That's one of the reasons you feel achy and sore. Your temperature's up, it's unpleasant, you get a headache. All of these things are the effects of those immune hormones. And so it becomes a question, is it a good idea to actually counter those, like by lowering your temperature? It may not be. If your temperature is getting very high, then it's a good idea to try and lower it. But when you're getting a cold and everything, rather tolerate those symptoms, because there's signs that your immune system is active. This can go wrong. There's no doubt this can go wrong. And in COVID-19 infections, many of the very dangerous effects appear to be because of overactive immune responses, which are producing really dangerous effects, very high temperatures, and some other things that I'll talk about now. So just remember that these things like your temperature are produced by hormones from the immune system. There may also be toxic effects from a pathogen as well that get added to it. All of these things are called pyrogens. Pyrogen means fire maker, produces heat. So the pyrogens are really important in inflammatory processes, but also in disease. So I just wanted to think about one thing. Have a look here and remember what we talked about. The effect on blood vessels here. The blood vessels here are dilating, but they're also becoming leaky. They're becoming leaky, especially to serum, to fluids, which that's what causes swelling here, right? But here it's just a localized effect. Just picture what will happen if we generated all over the body an inflammatory response. That can happen. It can happen, for example, if bacteria end up circulating in your blood. You can end up developing inflammatory processes all over your body. All your blood vessels begin to dilate. All your blood vessels begin to become leaky. Your blood pressure plummets because of that, because the diameter of the blood vessels is increasing. Your blood pressure plummets. And this is a special situation. It's not common. Don't lie awake at night worrying about it. But this is a situation, a very, very dangerous situation called septic shock, which is the result of an overwhelming inflammatory response. And that is part of what happens in the COVID-19, in severe COVID-19. People go into the septic shock where they develop this global inflammatory response. All the blood vessels dilate, or they all become leaky. There's all sorts of other effects as well, but those are the main ones that lead to this catastrophic loss of blood pressure, which is by definition septic shock. Okay, so just for the moment, remember what I told you about this cell called the dendritic cell. It's kind of wandering around in the tissues. When it is stimulated, it leaves the tissues, it goes into the lymphatic vessels, makes its way to the lymph nodes. We're going to talk about now about how the dendritic cell interacts with cells in the lymph node to produce this adaptive response. The adaptive response depends upon the lymphocytes. Remember what I showed you in the blood slide. They're small in circulation. They are a small cell with a big nucleus in the center. There are two kinds of lymphocytes that we're going to talk about. And these are the T cells and the B cells. T cells, I told you, they're mature in the thymus, this immune gland underneath the breastbone. The B cells are called B cells because they mature in the bone marrow. They're both produced in the bone marrow, but the T cells migrate to the thymus to mature. But that's where they get their names from. There are also, I will tell you now, there are two kinds of T cell that we are going to talk about. And you'll see why. They're called T helper cells and cytotoxic T cells. And you'll understand their exact function in a minute. So we've got a variety of lymphocytes, mainly in not exclusively, but mainly in the lymph nodes and in the lymphoid organs. And the dendritic cells especially are responsible for communication and for coordinating the responses of these T cells. So the dendritic cell is a tremendously important cell. There are four main characteristics to the adaptive immune system, and compare these always in your mind with the innate immune system. The first is the lymphocytes, the B and T lymphocytes. There is an enormous diversity of different B and T lymphocytes. The diversity is conferred by proteins which are embedded in their cell membrane. And it's with those proteins that they communicate to the outside and to one another. But there are many, 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 many different versions of those proteins, as you'll see. So first of all, there's a huge diversity of lymphocytes and of these receptors on the outside. The second really important thing about the adaptive immune system has to be very carefully regulated, because it is possible for the adaptive immune system to begin to act against our own cells. Remember that the primary responsibility of the immune system is to recognize those cells which are not self or no longer self, truly self. Those which have transformed into cancerous cells, cells which are pathogens, cells which are damaged or infected. These are no longer truly self, and they need to be eliminated. But the adaptive immune system must not attack our own cells. So it has to learn what is self and what is not self. And that's part of its adaptive ability. We call that self-tolerance. The adaptive immune system tolerates our own cells, but it reacts against anything which is not self. The third thing, which is important, and I'm not going to try and explain it quickly now, I'll show you rather in diagram form. There are there's a huge diversity of lymphocytes, but there's very few of any individual lymphocyte. So we'll have a lymphocyte which has a particular kind of receptor on the outside, but there's only a few of those lymphocytes drifting around. But if I stimulate that lymphocyte, whether it's a B lymphocyte or a T lymphocyte, if I stimulate that receptor, one of the first things that happens is the cell frantically begins to clone itself, begins to increase its population size enormously, very quickly. It starts to clone. And remember, its clones are all identical. So now we go from just having a few, 1, 2, whatever, with that particular receptor structure, we'll end up with many. Quite quickly, we'll end up with many with that receptor structure. This is a key feature of the adaptive response, as you'll see when we put it all together. The fourth really critical thing about the adaptive immune system is the processes, its response, for example, to a pathogen or to infection. These are very complicated processes. They take time to make their way. The innate immune system reacted immediately because everything's pre-programmed. The adaptive immune system is different. It goes through a lengthy series of complicated processes. It takes 5 to 10 days to produce a full-blown efficient adaptive immune response. So that is something of a disadvantage because the pathogen can go very far in those 5 to 10 days. Luckily, we have the innate carrying on its fight for us, retarding it, but nonetheless, an infection can proceed quite far and sometimes fatally far before the adaptive immune response really reaches its peak. So, but the thing about the adaptive response is it keeps memory. It remembers pathogen. It remembers things it has seen. It keeps a memory of them. And if ever it sees it again, it doesn't have to go through the 5 to 10 day period. It reacts immediately, very, very quickly. At second exposure, very, very quickly the adaptive immune response will kick in. And very often you won't even know that you've been exposed again. So that is key. That is one of the huge successes of the adaptive immune response. Once we have been exposed to something, a memory is kept. The memory, I'm going to tell you, does vary somewhat. Some things produce stronger memory than others. But nonetheless, this is a key feature of adaptive response. We keep memory. And that's the basis of vaccination. In vaccination, we teach the immune system about something before it ever sees it. So that if it does ever see it, it reacts immediately and very strongly and eliminates it. So get these 4 features clear in your head, and it'll make things explain the rest of the processes much more clearly. Then I will over and over again use this term antigen. And really an antigen is anything which will elicit a response from the lymphocytes, from B or T cells. And what is a little bit difficult to conceive of is just think of something like a bacterium. So a bacterium is really, really relatively large. And on its outside, it has many, 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 many different substances, proteins and glycoproteins and all sorts of things on the outside, which could elicit a response to B or T cell. So the whole bacterium is antigenic, right? But if I take that bacterium and I digest it up, I cut it up into little tiny pieces and I end up with an individual protein, that protein itself may be antigenic. Not only that, that protein will have small parts of it, small parts of the antigen, of that protein rather, which are antigenic, which can elicit a response. Those little portions that actually bind to the receptors on B and T cells to get a response, we call the epitopes. So the epitope is that part of the antigen which actually binds to a receptor. So you'll hear me use these terms antigen and epitope, and just understand that the epitope is that part of the antigen that actually binds to the receptor to cause a response. And even if I have a little tiny fragment of the whole thing, a little tiny fragment that has the epitope on it can elicit the response as strongly as the whole big bacterium, because that's how the lymphocytes see. They see through their receptors, so they only see that little tiny epitope. They don't see the whole thing. They only see the little epitope that they can interact with. And you'll realize now that those receptors on the outside of the lymphocytes, the antigen receptors, are hugely important because they are the signaling mechanism. They signal to the lymphocyte, tell it what to do, what the next step that needs to take place. Okay, so this is an important concept as well. Here are the lymphocytes. They, on the outside, you see this Y shape. You're going to get very used to this Y shape. This tells you immediately this is a B cell because its receptors has this shape. This B cell here only has one kind of receptor on it, okay? And this receptor is only going to react to one epitope, one very specific molecular pattern, antigenic molecular pattern. Can be quite small, and it's going to react actually there at these ends here. So this lymphocyte here only has one receptor. There are billions of lymphocytes generated in a week. Okay, we generate many lymphocytes continually from our red bone marrow. And very few of them are identical. So we've got a huge number, a huge diversity, all of them with different receptors on them. It's the same with T cells. They also have receptors. It operates a little bit differently, as I'll explain, but they also have receptors. And what we're going to be watching for, especially importantly, are those T cells that have the same receptor as B cells. And we're going to have to link these guys up. This is like match.com for lymphocytes. We're going to have to match T cells that have the same, will react to the same epitope. That's a very complicated, difficult thing to do. Why? Because there's billions of lymphocytes. There's billions of B and billions of T lymphocytes. But there's very, very, very few that share receptors. But the adaptive immune response has mechanisms to actually link them up, as we'll see. So go back to what I said about the cloning. And remember that though we have very few of these, if I stimulate this, very quickly I'll get this cell to clone itself and produce many cells all bearing the same thing. So that's a key thing in the adaptive immune system. If we want to stimulate the cell, get it to clone itself, then everything becomes matching up and everything becomes much easier. Okay, yeah, let me just, I'm sorry to flip around. I just want to make sure that I cover everything, even if I tell you on a different diagram. Right, okay. Oops, sorry. So let's have a look here, a little bit more detail about the receptor itself. This is a B cell receptor. And it has this classic Y shape to it. The receptor itself is embedded in the plasma membrane. Here's the plasma membrane of the B cell, and it's embedded here. And this is the molecular structure. We'll only do it in its really simple form. This whole side here, this whole part here, matches this part, is identical to this part here. Okay, so it's a mirror. This is a mirror image on the two sides. You'll see it has four protein molecules that make up the whole thing. There's these long ones here. There, those are called the heavy chains. Heavy only because they're big. They're the bigger. Then you've got these chains here, which are the light chains. And they're light because they're small. Okay, they're bound together. This is a disulfide bridge, by the way, in case you want to know. And this, as I told you, this whole side here is identical to that side. So what is identical to what becomes a little bit, you just need to get it clear in your head. This here, light chain there, is identical to that light chain there. This chain here, the heavy chain, is identical to this heavy chain here. Okay, but just have a look at the very ends, because this is the business end here of this molecule. This is where it is actually going to bind epitope, bind the antigen, recognize and bind it. At the end of each of these, there's a little region. It's called the variable region, and that's a very good name for it, because when these cells are produced in the bone marrow, these ends here can vary enormously from one cell to another. As they're generated, the body generates new receptors continuously, and they are new because the body makes these ends different. That's why these are called the variable regions. This is where all the difference arises. And this variable region here is not identical to that. This variable region is identical to that. This variable region identical to that. So just, it's complicated, but just realize that built into the system is an enormous variability. That's the huge advantage, because what this end is going to do is it's going to recognize antigen. And it ends up that our immune system, even if we present it with entirely new antigen, if we wait long enough, we generate so many different receptors that eventually the likelihood is that we're going to generate receptors which can recognize the new antigen. This is one of the reasons it takes 5 to 10 days for an adaptive immune response to be produced, because you don't necessarily have cells sitting around ready to recognize the new antigen. You may, but it may be that you have to generate the ones that match. But it's a random process. It's not directed. It's a random process. We're producing these cells the whole time with all of these different receptors. And if one happens to match new antigen that's come in, bingo, we set off the whole adaptive immune response. So just get the basics of this molecule in your mind. This Y shape, heavy chain, light chain. That light chain identical to that light chain. This heavy chain identical to this heavy chain here. And I will tell you right now that in actual fact, the antibody that we talk about the whole time, we keep on saying antibody, antibody, antibody. All that antibody is a soluble form of this receptor. This B cell, if this B cell was to produce antibody, the antibody it produces is based on this portion of the whole molecule. It just doesn't have this bit that's embedded in, and it's secreted as a soluble. The antibody is basically soluble form of the receptor itself, of the B cell receptor. And you'll see why that this is a very clever trick in the immune system, because it means that if I stimulate this receptor with a particular antigen and this cell begins to produce antibody, all the antibody is going to be against what stimulated it originally. That's why this is what is so clever about the whole system. Are you all right? Is everybody okay? If you're there? Okay.
Yes, I'm fine.
Hello, Shanice. Yes, I know you're always there. It's nice. Every now and again, it's nice to hear somebody's voice. It makes me feel like I'm not shouting into the void. Where was I? Okay. Let me just tell you right now, if I take an ordinary B cell and just sitting on its own, and it's got its receptors, right? And I stimulate that B cell strongly enough with antigen, I can actually get the B cell to start producing antibody all by itself. So, and this is what would happen. The B cell, you remember, the lymphocyte was a small cell with a nucleus that just about filled the cell. The B cell transforms radically once it's activated, and it ends up forming this enormous cell, which is called a plasma cell. And the plasma cell this would happen in the lymph node typically. We would stimulate the B cells and they would form plasma cells. And the plasma cells go into circulation, but they'll usually end up in the bone marrow, where they can live for quite a long time doing what plasma cells do. So this is what the cell ends up looking like. And what I want to point out to you, think back all those ages ago in your first biology classes where we did cell structure. Look at all of this in here. This is all endoplasmic reticulum. What does endoplasmic reticulum do? It produces protein. It processes, and produces mature proteins. And this is why this cell has become enormous, and it's become filled with endoplasmic reticulum, because this cell has become a dedicated antibody factory. And the antibody that it's producing is a soluble form of its unique receptors on the outside. So this is the fate of the B cell once it's been correctly stimulated. It starts, first of all, it clones itself, divides many, many, many times so that there are many of that specific B cell. And second of all, they transform into plasma cells, which begin to pump out antibody. And remember, the antibody they're producing is specific. It's specific to the antigen that stimulated the B cell. There is a problem, though, and that is if I do this directly, if I just take a B cell and I stimulate it with enough antigen or whatever, and I get it to transform like this, the disadvantage of that is that first of all, the B cell forms very poor memory. They don't form efficient memory. And second of all, there are some disadvantages to the antibody that it produces. I don't want to go into enormous detail about it, but as adaptive immune response proceeds, the cells get better and better and better at producing good antibody. And if you just stimulate a B cell by itself, the responses are not that strong. It can be, there are instances, especially in case of bacterial infections, where it is enough, where this kind of response is enough, but it won't produce a good memory, immunological memory. To get the really refined, full-blown adaptive response, we need to pull in the activity of a whole lot of other different cells. And okay, let me, first of all, I'm sorry, this little diagram is just a summary of it. First of all, remember what I told you, a pathogen may have many different epitopes that are antigenic. Okay. The B cell only responds to a particular epitope. So here's a B cell. It happens to match. And this is the kind of system that I've just told you about. This is a B cell by itself being stimulated. And it's bound to, it's the matching epitope there. We say it is cognate. So it's bound to its cognate epitope there. It's got stimulated, frantically clones itself, and it begins now to generate antibody. And the antibody is simply a soluble form of this receptor. That's the clever trick, because this soluble antibody now goes into solution, percolates all through the body. It percolates with all of that interstitial fluid that leaks out of the capillaries and everything in the lymph. It bathes all of our cells, and wherever it encounters the antigen, it binds to it and begins to inactivate it. How? We'll find out a bit later. But here's the B cell cloning itself, all of those clones producing soluble antibody, all of that antibody going into solution. But there's something else to remember, and that is this pathogen may have many different epitopes. So it can actually stimulate many different lymphocytes. And so when we get a response, an antibody response, we often see antibody to many different epitopes being produced because different B cells are being stimulated. So that's all that this is. Here we're just seeing a simple example where 3 different kinds of antibody are being produced to stimulation. Okay, so in order to understand how all of this is coordinated, and how all the different lymphocytes and everything is in the lymph node, especially when lymphoid tissues begin to talk to one another. I need to introduce you to another molecule, which is called the MHC molecule, Major Histocompatibility Complex molecule. That's its full name. All cells in our body carry MHC, but there are two kinds. And the one that we are most interested in at the moment is called MHC Class 2. And that is only borne by a few cells. And here are three of them. They're the macrophages, dendritic cells, and B-cells. They carry this special molecule, this MHC Class 2. And what MHC Class 2 is able to do is it's produced inside a cell which has ingested a pathogen and has digested that pathogen and broken up all of the antigens as small as possible. And the MHC molecule binds it and carries it to the cell surface, and it displays the antigen. It displays an antigen to the outside. And it does, these cells do that so that they can communicate the information to one another. So this is a special situation. Okay, the cells actually respond to the presence of antigen by ingesting it, digesting it to small portions, and exposing it on their cell surface with MHC Class 2. The three in particular that we are interested in are macrophages, the dendritic cells, and the B-cells. B-cells can also do this, and it's a critical step that we need to explain. Okay, so we've seen already B-cells can respond directly, but that is a rather weak immune response. Instead, we're going to pull in these cells especially. I'm not going to talk much about the macrophages, how the macrophages interact. It's another part of the story we won't consider. We'll just consider the main thrust of the story, which is through the dendritic cells. So dendritic cells wander in your tissues. If they encounter pathogen, they have the unique ability, which macrophages do not have. The dendritic cells are able to recognize the presence of virus and digest it and display it, as well as bacteria, and fungi, and all sorts of other things. The dendritic cells, these cells can display many different antigens, many, whatever they have encountered. But the dendritic cells, when they encounter something in the tissues, they immediately move to the lymph nodes. And they do so because they need to start talking to other cells, especially to the lymphocytes. They display the antigen on their surface for a specific reason. And that is that they are going to interact with lymphocytes with the matching receptor to the antigen that they are displaying. So here's a basic explanation for you of, remember, I've introduced you already to the MHC molecule, and we are talking about here about a particular class of MHC called MHC Class 2, which displays antigen from ingested pathogen. This cell here, it says host cell. This cell could be a dendritic cell, or it could be a B cell. Both of them would do the same thing. Ingest pathogen, digest it, and then expose small fragments of the antigen on MHC. The reason that they're doing this is that this other lymphocyte that we've already mentioned, the T cell, cannot see antigen directly. The B cells can. B cells can bind directly to antigen and respond. T cells cannot. They have to see fragments of antigen on MHC. No matter what kind of T cell you're talking about, remember there are two, helper cells and cytotoxic cells. No matter which one, they have to see antigen in this form here, bound to MHC. This is just a diagram of what what the T cell would see. This is MHC on the outside, and this is antigen here bound to the MHC. MHC will display many kinds of antigen. It's in non-discriminating, as long as the fragment is the right size. Basically, it will display it. Okay, so here's what our situation is. Present especially in the lymph nodes, there are many different B cells, and we need to stimulate those B cells, but we must be careful only to stimulate B cells which have a receptor to particular antigens. So here we've got a new antigen entering. Let's just say it's in circulation. In the lymph node, a critical step is that the B cells bind to their specific antigen. Here are different kinds of B cells. Each of them has a different receptor, as you see. Only this, in our diagrammatic example, only this one is going to be able to bind that epitope there, okay, and be stimulated. When that happens, as we've already learned, the B cell will start to divide. If we stimulate it strongly enough, it will clone itself and it will start to produce the soluble antibody. But remember, although we can select this B cell line now and start to produce antibody, this is a very weak response. We need to strengthen this response somehow. And the way we're going to do it is we're going to pull in these other cells to do it. And but just remember, there is a critical step here, and that is that the B cells can ingest antigen and display it. This is very important because this is how they are going to talk to T cells. So this is true. This does happen, this simple line, just the B cells on their own. But we are going to start talking about how they interact with T cells. So let me just get some definitions for you. First of all, we are first going to have to realize that there are two branches to the adaptive immune system. I'm sorry, every time I talk about anything, I say to you, there are two branches. Now we're going to talk about this branch, and there are two branches of that branch. I'm sorry, it's just complicated. But there are two major kinds of T cell, and each of them drive or participate in a different part of the adaptive response. So the first kind of T cell we're going to talk about are the helper T cells, TH cells. And their major responsibility is to interact with B cells. So what they are going to do, the T helper cells are going to interact with the B cell. And now instead of the B cell just doing this all on its own, it's going to proceed through a path where it is strongly stimulated by hormones from the helper T cell. And it's going to produce a much better response than this simple response here. The second type of T cell is called a cytotoxic T cell. And I just need to explain a little bit about what happens when any cell starts to change. Any cell in our body, when it starts to change, it's the surface of the cell often changes because the cell begins to display parts of its own proteins. This is done through another kind of MHC molecule called MHC class 1. And what MHC class, all of our cells have MHC class 1. And we continually show proteins on the surface with MHC class 1. And the reason, one of the reasons for that is that this is one of the ways in which the immune system is able to distinguish self from non-self. As long as you're showing your own proteins on the outside, the immune system leaves the cell alone. Some of the MHC class 1 molecules start to show bits and pieces that don't really belong, either because that cell has transformed and become tumorous, or because it is infected with something, then the immune system sees that and will try to kill that cell. And that killing action is through these cells called the cytotoxic T cells, TC cells. They specifically recognize infected or tumorous cells and try to kill them. So we've got two arms to the adaptive response. The first is this arm here, and it's called the humoral response. Humors are things which are dissolved in the blood. So it's a good name. The humoral response is the antibody response. So the humoral response, these cells are producing antibody, which is going into circulation. But there's this other very powerful arm of the adaptive immune response, and that is the cytotoxic T cells. We take cytotoxic T cells and the immune system trains them to recognize cells which are infected. Cells which have been infected get killed by the cytotoxic T cells. By the way, one of the reasons that you get such a terrible sore throat when you have a throat infection is because your cytotoxic T cells are busy rushing around killing cells which are infected with virus. That's just one of the reasons. There's other reasons you get a sore throat, but that is one of the reasons. These cytotoxic T cells are tremendously important in our day-to-day living because they are one of the ways in which we survey for potentially cancerous cells in the body, but they also are part of an adaptive response to infection. Okay, so here is the basis of the story. We'll talk about it in skeleton form and then maybe amplify it a little bit. But this cell here, please write in on your notes, and we're going to write in here that this is a dendritic cell. This dendritic cell has been wandering around the periphery. This ingestion here took place somewhere, took place in your finger, right? And the dendritic cell immediately, it saw that there was a pathogen present. It ingested the pathogen, digested it, and started to display fragments of antigen on its surface. It displays it with this MHC class 2 molecule. And it does so because that is how the dendritic cell is going to interact with T cells. As soon as it ingested this and began this digestion process, the dendritic cell migrated from wherever it was to the lymph node. In the lymph node, there is a possibility that it is going to interact with a T cell. Remember this. In the lymph node, there are many, many different T cells. There are T cells with many different receptors. Each T cell has its own specific receptor for a specific epitope. They continually interact with dendritic cells. As soon as the dendritic cell arrives, it begins to interact with all of the T cells. Most of the T cells cannot interact with the dendritic cell. Why? Because they don't have a matching receptor. If they don't have a matching receptor, they can't interact with the dendritic cell. But if they have the matching receptor, they bind to the dendritic cell and they are very strongly stimulated by sets of cytokines which are released by the dendritic cell. And the T cell now begins to clone itself. Now from being a rare item, a T cell with a rare receptor on it begins to clone itself, and it forms a whole population of T cells, all with the same receptor. And the reason for that is that this T cell is going to have to find those very rare B cells that have the matching receptor. So here's the step that we need to understand. The B cell, remember I told you, in much the same way as a dendritic cell, really. A B cell, when it encounters new pathogen, it will ingest it, digest it, and display it. It will display the antigen on its surface. This is the reason why. If in fact the B cell displays the same displays a receptor which matches the T cell receptor, they are able to stick to one another. So we've made a whole population of T cells with a specific receptor. The only B cells that they're going to bind to are those which have the matching receptor. And as soon as they do that binding, they release very, very powerful cytokines, which stimulate this B cell now. And the B cell now does two things. The first thing is it begins to frantically clone itself, form plasma cells, and release antibody, specific antibody. But the second thing it does is it creates population of mammary B-Cells. So now what we've done is we've produced a full-blown adaptive response. We brought the information about the antigen. In fact, we brought fragments of the antigen to the lymph node. We've stimulated a specific T cell. This T cell now, the T cell now clones itself. This is a T helper cell. Those helper cells, they actively seek out B cells with the matching receptor there. There's the key. It's a lock and key system. It's this key only fits this lock. And once that lock and key fit together like that, this B cell is strongly stimulated by these hormones, begins to divide frantically into plasma cells. A whole big population of plasma cells move out into the body. Ultimately, they take up residence in the bone marrow. They live quite a long time, and they produce antibody. They produce lots and lots of antibody. And I will just tell you quickly, I won't tell you how it's done. It's quite a complicated process. But as this process goes on, the antibody they produce works better and better and better and better. They also produce these Mammary B-Cells. The Mammary B-Cells are very, very long-lived cells. They can live your entire lifetime. And look what they are doing. They have the same receptor, but there are more of them. But not only that, when they see, they can see the antigen directly. If ever they are re-exposed, they have missed out this entire step and they jump straight to here. So the memory cells they can start operating very quickly, and they will start, even if it's years later, when they are stimulated, they produce the very best antibody very, very quickly. So, and if this is their second exposure, obviously, they wipe out a pathogen that they saw before. So, and that is how the memory works. Okay, I'm going to stop there. If there are any questions? I know this is complicated, okay, but think of it like a story. And then I also want you just, you see this figure here. If you could please print this out before I get to it, so that you can follow along. Because this, if you understand this figure here, you will understand everything that I've told you. This figure gives you the complete story, complicated though it is. All right. Okay, then I'll see you guys on Monday. I'm doing all of these immunology classes live, just so that you have a chance to ask questions.
Okay, enjoy your weekend, Professor.
You too. Stay safe and well. I am completely vaccinated now. So you can just look at me and I'm just a big bag of antibodies to COVID. So
I was talking to Professor Bacho about vaccinations yesterday during the tutoring session, and she was kind of convincing me, like just talking about her experience to get the vaccine, because I was kind of skeptical about it. But I was relearning about the immune system. It's making more sense.
Okay, good. I'm glad. Yes, I had both of mine. I've had both injections, and I had the second injection. I had a little bit of a sore arm. It was like having a tetanus shot, but I had no other reactions really to it at all. And it was gone a day or two later. It was completely gone. So yeah, I'm very happy. I feel much safer, much happier I still wear a mask when I'm out. But I feel much happier just being able to go out in the street or get on the subway and everything. Yeah, it's a good idea to get it. Okay. Then everyone. Yeah, I'll see you on Monday.